In the next couple of videos we will go over a number of tools that can be used to analyze NGS data in order to get you acquainted with using Galaxy to run analyses and the different data forms that are used during these analyses. The tools that will be used all come from this workflow. When a sample is sequenced, you get a raw image file from the sequencer. As you can run different samples in the same run, these samples have to be separated afterwards. When the sequencing is done on the MySeq, this demultiplexing is done for us by the instrument, so we get separate FastQ files for each sample. The samples then have to go through quality control to check whether the run yielded high quality reads, this is done with FastQC, and to trim the reads that do not meet a certain quality threshold. With the clean FastQ files, you can then identify the contents of the sample with Kraken and continue with mapping and de novo assembly. In this video we will go over the quality control with FastQC. Before we can start with quality control, we first have to import our data. This is the default Galaxy screen, and we have an unnamed empty history here, which I'm going to rename assembly, as we'll be using this throughout the other videos again. So now it's called assembly, and we'll be importing data from the shared data libraries. So we just go to shared data, the data libraries, and we'll import the first training set from the Galaxy training, select all files, and we'll import it to our history called assembly. So just click import, and the files will now have been added to our history. To run FastQC, either we know where FastQC is located, which is in NGS QC and manipulation. So we can scroll down until we find FastQC over here. Or if you want to do it a little bit faster, you can just search for the tool here, type FastQC, and we'll only get the tool as a result. When we click on FastQC, this page opens. So here we can specify our input file that we want to have quality controlled. I'll select the first one here and just click execute. When you execute, these files will be added to your history. These are the output files. And you could see that first they were gray when they were just added. Now they're yellow. This means that the program is running in the background. Once they will have completed, they will turn green. If you suspect that this is taking too long, you can always refresh your history so that you get the latest status update, but normally it should refresh itself instantaneously. There's one more thing I want to show you. If you click on FastQC and you get to the input page, you can also select multiple data sets by clicking this icon here. Now you can select multiple input files at once. This does not mean that these files will be analyzed together, but a separate FastQC instance will be launched for each dataset separately, and you will get separate output files for each input dataset that you specified. So if we click execute now, we will get 4 instead of 2 output files. And now that we have these output files, we can have a look at what's in them. To look at them, just press the eye icon on the file that you want to examine. And for FastQC, you will get an HTML report that starts like this. On the left-hand side, you see a summary of the results. A green check mark means that everything looks normal. A yellow exclamation mark means that the results deviate slightly from what is expected, while the red cross means that the result appears to be very unusual. It is important to know that even though the results appear abnormal, that this might be perfectly normal for your sample. Only when you know your sample and what was done in the wet lab, you can interpret these results correctly. As we will see, the 2 red crosses in this output will be perfectly normal for our sample. The first metric is the per-base sequence quality. Here we see a graph with box plots showing the distribution of the quality scores of all reads. The blue line represents the mean quality of the reads of a specific length, and the small red line in the boxes indicates the median quality. As we can see here, we have some longer reads with lower quality that should be removed. Next we see the per tile sequence quality. This is actually a view of the flow cell in the instrument. If you find some large blobs here with low quality, it indicates that there was something physically wrong with the sequencer. For instance, there might have been some dust preventing the camera from focusing correctly. An image like this is nothing to worry about, although you see the exclamation mark. Here we see the per sequence quality score. A graph like this is perfectly normal, as also indicated by the green check mark. A bump in the beginning of the graph, indicating that you have many reads with low quality, could indicate some kind of systematic problem. Next is a per base sequence content which failed the quality check. This is because we have this non-random distribution of A, C, G and T in the first part of all reads. If the DNA would be cut completely at random before sequencing, you would expect approximately the same percentage of nucleotides over all reads at every position, somewhat like the rest of the graph. What we see here is an artifact of the Nextera XT kit that was used in the library preparation. This enzyme does not cut completely at random, but has an affinity for some sequences. Therefore, at the beginning of the reads where the enzyme has cut, has a bias towards those sequences, and this is visible here. So, in short, for this data, these results are perfectly normal. If you would see these results in a completely randomly cut sequence, then there would be an issue. Here we see the per sequence GC content. Many samples will not pass this test and show the yellow exclamation mark. The most important thing to check here is whether the peak of the GC content is at the position that you would expect from what you know about your sample. Small deviations from the theoretical distribution are normally not an issue. If you do find the peak at another location than what you would expect for your species, or when you see one main peak and a minor one somewhere else, it's a good indication that there was a contamination or that the sample was mislabeled. The perp base N content should be very low. In some sequences, the software will replace an uncertain call with an N. If you would see many N's in this graph, it means that the base caller was unable to call valid bases. The sequence length distribution shows how many reads you have of a certain length. If you would see a bump in the left part of the graph, it would mean that there were a lot of reads that did not extend to the maximum read length. The small area under the curve here is created by the fact that the Nextera XT kit also creates fragments that are shorter than the maximum read length. Therefore, it does not indicate a problem with the sequencer, it just means that the fragment could not be sequenced any further as it was not long enough. In a completely random library, most sequences will occur only once in the dataset. If you would see a high level of sequence duplication, there might have been a PCR bias so that only a certain fragment has been amplified. So if you see that the graph is high on the right side, or if you see any spikes in the right, this indicates an enrichment bias. Do keep in mind though that the binning size is different towards the right, so it's normal to see small bumps as these are the result of differences in bin sizes. The graphs that we saw up to now are all from a good quality dataset. Now we'll go over some graphs that you might get if something is wrong with your data. In this first graph we see that the per base sequence quality is really bad. The quality starts dropping really fast and you will lose a lot of data once you're going to filter on base quality. In the next graph we see that we have a very bad quality per tile. There are several cells that have a quality that is much lower than the average. These are indicated in the red color. Next we see a graph of the per sequence quality, and here we see that the largest number of sequences have a mean Phred score of 13 to 14. The graph also shows several peaks indicating that something went wrong on a subset of sequences which might be caused by an issue with the flow cell or imaging. In this graph we see that the sequence composition is almost exactly the same for the first 20 bases, and then it starts to average. This can be the result of techniques used to fragment and amplify the DNA before sequencing. In case of amplicon sequencing, for instance, this is something you might see if the PCR adapters are being sequenced as well. This might not be a problem per se, but there might be an issue with read quality further down the read. Illumina uses the first cycles of a sequencing run to determine the clusters. When all clusters have the same composition in the first bases, some clusters might be merged based on these sequences. When the sequences start to diverge later on, the sequence will have problems to identify the correct bases in these merged clusters and therefore result in lower quality calls. Another bad example of the sequence composition can be seen in this graph. This graph is a result of adapter dimers that were formed during the library preparation. Normally, the procedure to create the library should prevent adapter dimers to be formed in the library, but if something goes wrong, you will get an output like this. Now, this is an extreme case, but in milder cases, you will still be able to identify a pattern like this. The same adapter dimer issue will create a GC content graph like this. You see a huge spike that is created by a large number of reads that come from the adapter dimers. You might also get peaks like this when your single organism sample is contaminated with another organism with a different GC content. Another example is seen in this graph where we see multiple small peaks. In this graph we see a large number of sequences that have been duplicated. This can be caused by a technical error during the library preparation, but it can also have a biological cause. The graph that we see here is caused by only sequencing the small bacteriophage phi X on a whole lane, resulting in each position being sequenced thousands of times. So in this case it's a biological effect and not a technical one. This is once again a reminder that you need to know exactly what you're sequencing in order to correctly interpret these graphs. If you want some more information on FastQC and how to interpret the different metrics it reports, please go to their website where you can find an excellent manual that explains everything in much more detail. This concludes this video, and in the next video we will continue the analysis of our sample.